Well, this is John Carroll. I'm the editor of Endpoints News. I'd like to welcome everybody for coming out today. We had a big audience sign up for this one, and I think it's pretty obvious why. Gene therapy is the big thing that's going on right now. There's a tremendous amount of attention on gene therapy, everything that's been going on. And a lot of different issues that have come up that I think we'll have a lot of fun discussing today. We're joined by a panel here that includes Kathy High, the Spark co-founder and new president of research at Ask Bio as of, I think, yesterday, Kathy. Sandy McCrae, the CEO of Sangamo, Dave Lennon, the president of Novartis, and Jim Wilson of the University of Pennsylvania. But first, before we start the panel conversation, our sponsor today is Ignite, and we have Alok Tai from Ignite to offer us a few minutes commentary about the work that they do. Hello?
Awesome. Thank you so much, John, for the intro and the opportunity to present here today. We're really thrilled to be both a partner for Endpoints as well as have the opportunity to sponsor such a critical topic and event. As John mentioned, I'm the Vice President of Life Sciences here at Ignite. Ignite provides software to help companies manage documents and data, specifically in the life sciences domain. And so really to kick off this discussion, I'd imagine many folks in this industry have been experiencing, especially over the past year, a tremendous amount of complexity and change in terms of how they operate and how data is managed in their organization. I think what we're seeing is that as individuals are either working from home or outsourcing and working with external collaborators or trying to bring novel medicines to market like gene therapies, there's a tremendous amount of complexity, especially around how they handle data, regulations, and collaboration. And so in this sort of environment and circumstance, this is where Ignite, I think, as a company, wants to come in and help support organizations like yours. And so what Ignite does is we provide a unified platform specifically for life sciences documents and data. So whether you're looking to help facilitate exchange and collaboration internally or externally, help ensure compliance with, say, FDA-oriented regulations and reg control documentation, or think about this new world of data privacy, Ignite can be a really good partner for companies of looking to accomplish those sorts of goals. And so as a business, we're really fortunate to have thousands of customers as an overall company, 500 to 600 of which are in the life sciences domain specifically. And as a company, we've been around for more than a dozen years, and have enjoyed close collaboration with many of the companies you see on the screen today. And so at the end of the day, the approach that we take is to really help understand some of the specific challenges companies like yours are facing. And what we've seen across our large customer base and across the way the industry is operated is that these challenges really amount to three different types of issues. The first is managing a suite of regulated activities, whether it be manufacturing, clinical trials, regulatory submissions, et cetera, inside or outside your organization. Second is facilitating collaboration with a myriad of different partners involved in the drug development process, as well as, again, this really unique set of data privacy-oriented regulations like GDPR that are also impacting how organizations prosecute their drug development activities. So to help support companies, we've designed an industry-specific platform called Ignite for Life Sciences, which has what we call the 3 Cs. The first C stands for compliance, to help you meet those FDA-related regulations. The second C stands for collaboration, to help you facilitate internal and external collaboration with critical entities. And then the third C is for control, to help you comply with those additional regulations and have true master over your data. We've been really fortunate to collaborate with some really promising companies like Therapeutic, based on the West Coast, and really help support them in their growth from a very young company now to a publicly traded one as well. So, with that, would love to engage in a deeper dialogue with each of you offline, and hope you have both an enjoyable JPM as well as I know I am looking forward to today's gene therapy related conversation. So, John,
Okay.
over to you.
Well, I appreciate that, and I appreciate you sponsoring this conversation. So let's open it up. But first, before we do, I just wanted to tell the audience there's a facility on this program for you to ask questions. So if anything comes up to you that you'd like to present a question to the panel, please go ahead and submit that, because we'll be screening the questions as we go through the conversation, picking out a few that we can get to before the end of the program. And I'd really encourage you to do that. If there's somebody you'd like it to direct it to, just say this is for Jim or Sandy or Kathy or Dave. Just let us know. Otherwise, we'll just open it up to the panel. So with that, we're in a very unique situation for a new technology. Everyone here has been working with gene therapy in one fashion or another for quite some time. I think it has exploded in terms of the amount of activity that we've been seeing going on. We've been seeing a multiplicity of programs, a multiplicity of companies. We've been seeing a variety of different programs going after the same disease. We've been seeing a tremendous amount of activity at a time when IPOs and venture capitalists have been funneling billions of dollars into the industry, in part because of the attractiveness of what's going on with gene therapy right now. And obviously the patient populations must be quite engaged in terms of what's happening, because often these are very, very serious ailments that are being addressed, and something where they'd like to see something come up on a once and done kind of an approach. At the same time, last year, I think we've seen a tremendous number of setbacks. And I think it's raised a lot of questions related to safety, raised a lot of questions related to durability, raised a lot of questions in terms of regulatory oversight. So lots to get on. Now, I want to start with Jim, because Jim, you've been doing this from the beginning. And and you were one of the first ones to send the signal that there are maybe some safety issues, that there are safety issues that we need to address. So where do you see this right now in terms of the safety issue and in terms of being able to resolve that with the technology that we have?
Yeah, the decision that I made and many others in the field to focus on AAV for in vivo gene therapy, at least from my perspective, was based on the notion that it would likely be safer than some of the other delivery systems. And in fact, everything that I've tried for in vivo delivery, adeno, non-viral, retro, lenti, AAV was and remains the safest delivery vehicle that I've ever studied. And in my view is incredibly non-pro-inflammatory, and the safety profile is amazing. With that said, when you push the dose and use novel delivery approaches, you're going to eventually reach dose-limiting toxicity. So I was a little surprised that people were surprised that a biologic could have dose-limiting toxicities. So, with that said, once we understand what they are, I'm sure there are ways to mitigate those, and I think that's where we are now. That the field was pushing the dose and pushing the envelope, and started to see some of these toxicities. But again, that in no way, from my point of view, indicates that the platform itself is inherently pretty darn safe.
Well, typically, whenever you see a dose limitation issue come up with any kind of therapy at all, though, it raises questions in terms of durability, particularly for gene therapy, and the effectiveness of what you're getting across. Maybe we could open this up a little bit. I mean, in terms of whether or not what everyone else here is seeing related to maybe AAV, or also safety in general, the kind of different issues that have been coming up. Sandy, how about you?
It's interesting to hear this discussion, because I spent nearly 20 years doing drug development with small molecules and monoclonals, and they always have dose limiting toxicity, always. And the idea that gene therapy would be easy, I think, has passed. that time passed. Like all medicines, you've got to choose your dose wisely and balance benefit and risk. Actually, one of the challenges we have with such a broad portfolio is things don't die in the IND phase. And so you have to prioritize, not based on what you would do with a small molecule around toxicity, absorption, distribution. So I think it is relatively safe compared to most medicines, but we have to choose wisely which diseases we go for, so as we match the benefit and balance the benefit and the risk. I mean, we stand on the shoulder of Jim and Kathy and all the work that they've done with AV, and we've learned to go prudently and carefully. And if you do that and go into the right diseases, I think there's a way forward. I read yesterday about hypercholesterolemia being edited out. That may not be the right benefit risk at this time. That feels like a disease that is addressable by other things. And so I think we need to choose where the patient has no option or the disease is life-threatening or severely disabling, and then go prudently as we raise the dose of the virus.
That was an animal study, of course, that was done in that particular instance. I think we're still some years away. That gets into the whole issue of broadening out a technology that seems to be tailor-made for very small population types of diseases where the price is going to be extremely high. If you go into cholesterol or something like that, in terms of heart failure, you're talking about mass audiences where the whole pricing thing has to change as well. So Kathy, I mean, what's your take here in terms of the safety issues that have been coming up? Is this something that you think is readily fixable, or do you think that there's going to be some sort of more fundamental change necessary on the technology side?
Well, I think I would make a couple of points, John. One is just that to add to what Jim and Sandy have already said, you have to remember that right now there are many more trials going on. There are many more trials, there are many more patients. And so it is perhaps not too surprising that you're seeing more adverse events. That just is a function of the dramatic increase in the number of trials. I think there are some areas that we perhaps understand less well than we would like to. And one of those that I would highlight is, what are the characteristics of a manufactured AAV vector that may affect clinical outcomes? I think that's one of those issues that honestly we know less about than we might like. And I expect that we'll learn more in the coming decades. But I would really agree with Jim. I don't think that we're seeing showstopper problems. I think we're seeing problems that have to be evaluated very carefully at the clinical and molecular level. And people have to continue to work towards solutions, but I don't think we're seeing problems that can't be solved.
Well, let me follow up on that, because one of the things you are seeing a kind of, you're saying that the proliferation of companies and the proliferation of programs will inevitably give rise to problems, because drug development of any kind is not easy. You're always going to be running into different issues, but there does seem to be a commonality associated with high dose delivery of gene therapies, where companies tend to repeatedly run into the same problems. And I do wonder whether there's a reevaluation on it certainly seems to be a reevaluation on the part of the FDA, but a reevaluation in terms of overall just limiting doses to, going into this thing. Is there a recognition that there is a common problem, or do you think that each company is going to go at this their own way?
Yeah, let me just comment on that. I do think it's a platform problem. But with that said, there are many factors that would determine the therapeutic window. I think the story that has played out with Audentes, illustrates an important point that Sandy brought up, and that is they did have fatalities at a high dose for MTM, but they're off clinical hold now, presumably at a lower dose. And so the risk benefit there clearly justifies threading the needle on a dose. And I applaud the FDA for looking at reviewing the data in the context and allowing them to proceed forward in that really horrendous disease. But there will, Kathy had mentioned, referred to residuals, the way it's manufactured. There are various different components that would change where that dose limiting toxicity is. And I really behoove the sponsor to do whatever they can to develop a dosing schedule for which it would be, that there would be a therapeutic window. But in many of these situations, it is pretty narrow. The difference between where effect is and toxicity. One other point that is problematic is the relationship between dose and efficacy and dose and toxicity is not linear.
Yeah.
That when you reach that higher dose, it just shoots up both efficacy and toxicity. So understanding that would be really helpful. But there are programs that I suspect, such as the MTM program, that could go forward with high dose.
So Dave, did you have an initial pushback as it related to high dose safety issues?
I mean, I think, anytime you're looking at the disease, the FDA is determining what's the relative risk benefit that you're taking in the population, especially since a lot of these drugs are going into pediatric populations initially for genetic disease. And certainly you've seen recent guidance that where the FDA is asking to do adults first wherever possible in these cases and making adjustments. And I think it's a learning process. I don't see it as, we can say the bar is raising, but I mean, I think it's just collective learning and the application of that learning in a more determined way. I do worry a bit, maybe about a bit of the gold rush mentality of early investment and the proliferation of companies, because the tools and the platform is actually quite robust, as Jim and Kathy pointed out. And that means it's also quite facile to go from concept to therapeutic and start to put it into clinic. But the reality is that set of barriers being very low belies the fact that there's a whole set of barriers for submission quality efforts, management of the programs, manufacturing, and other components that a lot of folks aren't coming to grips with early enough in their programs to be successful as companies.
Well, it's interesting because manufacturing is an issue, I think, throughout the industry. Some companies do it extremely well. Some companies don't do it extremely well. You run into a lot of issues with some biotech companies, I think, when the biotech companies don't pay attention to CMC at an early stage. And that tends to happen throughout the industry. I mean, it's not any particular, therapy or any type of therapy, but, the issue about manufacturing and working out manufacturing, where are the key challenges? Why are there problems associated with this? And what do the companies need to pay attention to?
And I'll start maybe first and foremost, we don't have full characterization of what we're actually dealing with as a product. So we have to understand that when you look at the complexity of an engineered viral particle, you're talking about orders of magnitude in difference versus small molecules and even biologics of the past. And that level of complexity and heterogeneity in our product and ability to actually analyze it is the first challenge for any company. And the tools and the analytics that go around that are still under development and being advanced. And so just the first challenge for any company is characterization of the product to make sure that you can actually measure what you intend to produce from time to time. We're currently producing commercial grade product every day, 24/7, 365 days a year. And in that is tremendous learning. It's tremendous learning on yield, it's tremendous learning on consistency, on product characterization, and on developing and improving our analytics. And I think it'll take a number of products getting to that commercial stage for the industry to really be able to address that characterization challenge. I mean, you then can get into issues on scale and other things, but I think fundamentally we still don't necessarily know what we have sufficiently to be able to predict consistency from batch to batch, product to product, and to be predictive in our ability to say how that's going to impact how these products perform in the clinic. We're getting a lot better, and I don't want to be all gloom and doom about it, but it's still a lot of work that needs to be done.
Let's open that up. Kathy, obviously AskBio did a lot of different work in terms of CMC early on, bought some things and moved on that. So you must have paid a lot of attention to that at Spark as well. So what's your perspective here?
Well, I would really echo the things that Dave has said. And as I said earlier, I think one of the areas that the field really has to work on is understanding how the characteristics of the product affect the clinical outcomes. And I think that is really in a nascent stage for this field, partly for the reasons that Dave mentioned, that, analytics are in a continuous process of improvement and development. And, we are all learning best practices for analyzing products. And yes, that was a very important focus for Spark. For the BLA for Luxturna was analytics. And quality systems and bringing all of those to commercial standards. And I completely agree that the more experience the field has with that, the better. One of the things I really like about AskBio's manufacturing platform was that they did find a way to move beyond plasmids and are using essentially synthetic DNA as a starting reagent. And so you don't have to, you're not in these long queues for getting your production plasmids, and you don't have to worry about bacterial sequences and so forth, and antibiotic resistance genes potentially packaged in your final product. So I think we'll continue to see innovations like that in the field, and that it's going to lead to safer and purer products overall.
So,
John, could I just comment on that, because I hear criticism of manufacturing for AAV. There's concerns about it, and my perspective is that the field became real when a few academic institutions made the commitment to produce vector for clinical trials. It was CHOP and Kathy and LCA2, nationwide for SMA, and St. Jude's for hemophilia. And God bless them. With the human data, this became a field. But no one was going to invest in manufacturing until there were clinical data. And so there, we had positive results based on those early sort of manufacturing processes, and now we're sort of playing catch up. But we're going to play catch up. But maybe I'm getting a little defensive here, but I think the field, there was no way that we were going to get ahead of manufacturing before we even knew it was going to work. So I think it's just a matter of a latency period in places like Avexis and Spark and others, and it will become professional. But we are catching up, along with the fact that, as David mentioned, we really don't have as good idea what's in our preps that we ultimately are going to need to know what's in our preps. So that's going to be an evolving landscape.
It's a great point that Jim makes, and sorry, John, that in many ways the clinical results have been so spectacular that we're doing a lot of development in reverse. Yeah. And so, you're starting with these fantastic results in some, 5 patients, whatever it is, and then you're kind of building the program backwards to generate all the data you need to justify moving that forward in a safe and reliable way. And that's a big challenge for the industry. And, it creates a lot of discomfort at the FDA, but also a lot of challenge for patients and families that want access to these therapies as quickly as possible, see fantastic results in 2 or 3 patients, and then are still waiting years to get there. And, that's something we have to close the gap on.
So,
go ahead.
Can I just riff on a number of the things there? So manufacturing is so important. We've just completed our manufacturing site in-house, and we did that because we want the scientists to be beside the CMC people, so as the process can be controlled and understood as you go. These are rare diseases, so if you're doing a trial with, 10 patients in it, it's really hard to correlate why that patient had one result and this one a different one, and why this one had an abreaction. So it's an incredibly difficult correlation. Until we get more patients dosed across more trials with more vectors, will we really understand this. And then when we give the virus, we just give it by kilogram. Is that the right way to do it? That feels that it isn't, a 200 kilogram person's liver is probably about the same size as a 100 kilogram person's liver. And yet we give them double the dose. We need to find ways to correlate how much virus we give to the patient in front of us. And then there's one last one I think is driving the problems that we're seeing, is the concept that this is the patient's only chance. So one of the most interesting discussions we have with the agency is what's the starting dose? Because the patient doesn't want to start at a low dose that is not going to be effective. The patients talk about the hero dose, which is they're taking one for the team, and they're taking a dose that they know the next cohort or the one after that will be the one that's effective. And so one drives up to an E number that is higher because they can only get it once, they can't get dosed again and again. And that must get us closer and closer to the to the dose limiting maximum efficacy dose. And I think that's part of the reason that we will see some of the problems and see more of them. But one last thought about manufacturing. It took us about a year, 18 months to build our manufacturing site. That was easy. What's hard is finding people to staff it, and people who staff it and have expertise, because it takes many years to train them and to get them to be good at it. And the commonest call I get from a headhunter is, do you know anyone that could run my manufacturing? Because there's just a shortage of these people out there. And there needs to be, perhaps in universities, a real education drive and a training drive, so as we can get people that can do the CMC, because that's the bit that's the most important, getting your drug registered.
Kathy, you laughed at that. You recognize this issue.
Yes. Are you talking to me? Yes,
Yeah,
absolutely. I remember when we were starting our clinical grade production facility at Children's Hospital of Philadelphia many years ago, it seemed like everybody who came in and got trained in GMP production of AAV, you turned around, they were gone, they were upstream by industry.
Yeah. Yeah.
So it became a continual training program and coaching program and so forth. But boy, when they had a little bit of experience, they were gone. So I do completely agree with that assessment.
Dave, hard to keep talent?
Yeah, I mean, you'll see this this will only intensify. I mean, if you look at the number of companies that have announced new manufacturing sites, even over the last 3 months, it's incredible. And I think investors recognize that owning manufacturing is a key to moving these programs forward effectively. You can't wholly rely on CDMOs or academic institutions to own your destiny, right? In these programs, we have about 1,200 people today in manufacturing and quality, and I can tell you that it's challenged to keep that level of staffing going. We have continual programs and development programs now with various universities, and there'll be hotbeds of this activity across the country. And North Carolina certainly is one of the bigger ones, but Boston, New Jersey, and California will also be hotbeds of where this talent is. But as an industry, we do need to think about how do we manage the talent pipeline, bring that in holistically with universities, and create those opportunities. In the short term, I think it's going to be a pretty intense war for qualified talent. But in the long term, it's a great growth opportunity for an advanced technology industry particularly within the US, where most of manufacturing is focused today.
Okay. So in terms of redosing, I wanted to get into that a little bit, because obviously that has been driving a lot of the discussions related to some of the more advanced programs that are out there. You saw that, and also, in terms of, what people are looking for and how they're trying to prove that. I mean, you're in a very interesting situation right now with BioMarin, where they had their lead program, they took it out there, and then, they got handed it back by the FDA. There's a kind of an expectation right now that maybe the regulators are being a little bit tougher than had been initially expected related to this. It's interesting that the European EMA and the FDA have taken two different approaches towards the same data. One's looking for 1 year, the other one's 2 years from the trial. And it raises a lot of uncertainties at this early stage of seeing these therapies coming out and being commercialized. Do you feel like that there's a clear set of guidances from the FDA right now related to development, late stage development, and what they're looking for? Because some of these situations have been really quite stunning for the people that are involved, and investors as well.
Well, I'll take a crack at that.
Okay,
I will say this, for people who find the regulatory apparatus right now confusing or wanting, all I can say is, 20 years ago when we were doing the first gene therapy trials, it was a very different landscape. I mean, at least in the United States, we are no longer confronting this dual system of review by both the FDA and the RAC. And, very important, we now have these guidance documents first appearing in draft form in 2017, 2018, and now finalized. And I mean, I think that's been a tremendous step forward. I agree with people who are interested in more harmonization between the FDA and the EMA. But I have to say, at early stages, I think there's something to be said for regulators looking at things objectively and independently on two sides of the Atlantic and in other jurisdictions as well. So I feel that the regulatory apparatus right now is in a much better situation than it was when we first began.
Jim?
Is the question,
Do you think that the regulators have raised the bar? Do you think that they've changed the game at all? Do you think that this is something that still has to be resolved?
no, I just think it's the coming of age that programs that stood up a long time ago are now eventually reaching phase, registration trials and requests for market authorization. And that's where rubber meets the road. So we don't have a lot of experiences with that. I would say that one lesson that I've learned, again, going back to what Sandy had said, is the tolerance for uncertainty or risk, whether it's can you redose or how long is it going to last, is going to be driven in part by the unmet need of the disease. And I think a lesson, like Sandy had said, for the editing world would be, I would say, is stick with diseases for which there's significant unmet need. The agency will let you into the clinic, open INDs with limited subjects, but when you're talking about registration and commercial approval, I think the uncertainties about durability and redosing are going to play a much more significant role. And as we think about leaving or evolving out of monogenic diseases, which generally are more severe, I think it's going to be a hard transition for the field. I think we're going to have a lot more data, a lot more experience before we can go in that direction.
I completely agree, Jim. I think Peter Marks and Sieber does a good job. They're under-resourced and they're overwhelmed by the number of, it's a bit like the manufacturing. There's such an increase in the number of cases they have that they haven't yet got enough people trained to be able to do it. There's a phrase in French that's something like a happy man writes on white paper with white ink. So all the people that promised that they would get it through the agency in a year or 6 months with 10 patients worth of data, they hadn't actually done the clinical trial and proved the evidence that is now coming to fruition. And now there's black ink on the paper, and the agency rightfully says we want 2 well-controlled studies or we want this number of patients. My experience in doing due diligence for years is if you go back, the agency's usually said it and people have just looked at it with rose-tinted glasses and not always listened to what the agency says because it's painful. So I think we're learning with this field and we're getting there. But I think they're doing a good job. Try registering a type 2 diabetes medicine and you'll understand how accommodating CBER is.
I do think it's fair, though, to say to all of this that there is some level of frustration that exists within companies today. And it's on 2 fronts. I think one is that while the guidance may have been there, some of the assumptions folks take they look at parallels and say, why can't I do it that way? And some of it comes down to risk benefit or other approaches or unforeseen things that happen. There is this, I think, challenge around comparability between different production methods that has held up a lot of companies and has been very frustrating. As I think the BioMarin example is not a question of durability, it's a question of commercial process availability and the data related to that. And extrapolation around that has led to some challenge, I think, for the industry. And then I think you throw on top of that the pandemic. And I think the ability to get in-person meetings these days where you can really have dialogue is very, very limited. And that has really held up a lot of companies, I think, right now in getting some allowance, right, from guidance or getting some clearer direction around key questions that I think will continue to just hamper and delay some of the efforts over the next year.
But Dave, you had referred to, I thought it was an excellent point about it's almost too easy.
Yeah.
And I do think that that's kind of a problem, because I wouldn't want to be sitting in Wilson Bryant's seat or Peter Marks, because they get a lot of stuff that we don't know about that really isn't necessarily well done. But
It's very fair.
Is there any, I'm curious to get your perspectives, is there any kind of an advance, a breakthrough, a next gen approach to this that needs to happen? It seems on the manufacturing side that that's something that can be worked out, that you can put money into it, you can devote resources to it, you can get that worked out in one fashion or another. But is there some sort of a key issue that's yet to be resolved that's going to perhaps make it easier to get these drugs, to get these gene therapies through the process and into the market? Whether it's delivery or some other kind of aspect that you're looking at here that you want to see some real advances in, or you expect that everyone would need to see some real advances in before we get through this stage of where we're at? Sandy?
Do you mean the regulatory challenge, or do you mean just in general?
Whether it's the regulatory challenge or whatever, is there some aspect of the technology that's not quite ready for prime time, perhaps, or any other aspect of this that needs to be resolved? I mean, there's been issues related to delivery and new delivery.
John, it's delivery. That's the thing I worry about, is delivery. Zinc fingers, and I'm sure it's true for CRISPR, you do it in a test tube, it works every time. But getting it into tissues outside the liver safely is the biggest challenge we all have. AVs go to the liver, and the liver's become a very crowded place with the Alnylans, Modernas, and all the other standard gene therapy companies are all in the liver. The future has to be to get beyond that and get to tissues, and the dream is to get to all cells that has a gene that you want to correct. If we got there, we would be able to address so many other diseases. And I don't know, people like Jim are developing all these new capsids, but will they take us beyond the liver? Because majority will still go to the liver. And that's the challenge we have. If you could get us to the lung, we could do cystic fibrosis. If you could take us to the kidney, we could do polycystic kidney disease. It just is about getting to the right cell.
Jim?
Yeah, let's assume Sandy accomplishes it. Delivery. I think the area more than incremental advance is to be able to turn a gene on and off. Because the notion that you give the drug and it's on for a long time, and if something happens, you can't do much about it, does create some concerns about safety. And that was an area that was in vogue 10, 15 years ago and sort of fell out. For very severe diseases, that may be less of an issue, but looking sort of, John, a little bit beyond that, whether it's a kill switch, they call it, or an inducible on or something. I think if that really was feasible and work in a robust, reproducible way, that it would change the risk, benefit, and value proposition of gene therapy.
I think building on what Sandy and Jim said, I think I agree with what they've said. I think in addition, right now gene replacement technology is largely a positive enabler, positive therapeutic, which has opened up a whole world of treatments that don't typically exist. But the vast majority of drugs are inhibitory. The vast majority of diseases are about dysfunction, and the ability to introduce inhibition into the disease pathways, I think, is one of the bigger breakthroughs that the industry needs to have to get to move beyond genetic disease.
Kathy?
And I would just add that if we can understand a little bit better what are the drivers of transduction efficiency, so that we don't have to give a dose that has more viral particles than all the cells in the liver to transduce 5% of the cells. You know, I would be interested if we could make some progress on that question. And then I think, you know, we're really at the beginning of learning a lot of things clinically in terms of, you know, what are the drugs that are not compatible with being on AAV or getting an infusion of AAV? What are the characteristics of a population with a complex acquired disorder that are going to make some people susceptible to a gene-based approach of one type, but not, another group of patients who will never respond to that. So I think, you know, there are aspects of clinical medicine that need to be understood a little better as they relate to gene therapy.
I'm curious whether or not, well, I'd like to get your take on the price of gene therapies. Obviously, there's been a tremendous amount of discussion for how much you charge for a gene therapy, and the focus that is on that, whether that's sustainable or not, whether this is kind of the initial first generation kind of an approach. Is reimbursement, is the cost of gene therapy an obstacle to making them? I mean, obviously, it's driven a lot of new technology. It's driven a lot of work in the field. It's driven a lot of companies and people to get into gene therapy. I'm kind of curious whether there's a bigger reckoning yet to come as it relates to the cost of gene therapy and whether that's going to change anytime in the near future. Dave, how about you?
So I'd love to start on this. I mean, I think when you look at the value of a therapeutic, objectively, gene therapies are some of those valuable medicines that you can give because of the benefit that they provide, whether it's curing blindness or extending life. And I think that in that framework, the prices we have today are justified for the benefit that those products are providing. And that's the approach we've taken. It's also underlined by a commitment that we have as largely in Western societies to have deference to taking care of people with rare diseases who are largely underserved. And this extends to things like enzyme replacement therapy and other rare disease treatments that have always actually cost very high levels for these very small populations. And it's that model that has driven investment in rare disease and a lot of real great advances for these types of patients. And in my mind, when I look at that and look at the overall health economics of that, it is sustainable in that setup. Now, if you said, hey, we're going to have a gene therapy for Alzheimer's, would you charge the same amount? Absolutely not. I mean, it's just not the same value proposition. It's also not the same return for the investments that companies would make. And you would make the adjustments necessary to make that business model work in that situation. And that's, I think, one of the beauties of the system that we have that is largely driven by the US, which, you know, it relies on a capitalistic approach to solve these, to apply innovation to business settings. And I think that the framework that has been established for this, particularly in the US, but also in Europe, is really beneficial to patients with rare disease and beneficial to companies and beneficial to innovation.
Well, let me start. Did somebody else want
to? It's an interesting analysis because you can do it two ways. You can say, you know, a patient with hemophilia, they use, I don't know, $6 million worth of factor in a lifetime and $30 million worth of healthcare in a lifetime. And therefore, if I'm going to give them something that takes all that away, that's value saved to the system. But you can also do a calculation that says it takes, it's 100 or 200 million at least to develop one of these drugs by the time you've done all the manufacturing and the clinical trials. The cost of goods, you can understand that. The price you can charge without getting people, without having an abreaction a certain amount, and you're left with a prevalence as the thing that decides whether the commercial enterprise will ever be successful. And although there's 7,000 rare diseases in the liver, there's only about 10 or 20 of them where that equation makes sense. The majority of rare diseases are so ultra rare that you will never even be paid back what you paid to develop it. And a great credit to Peter at FDA and the people at NIH. They're trying to find a new way to deal with this, almost like a foundation where we each contribute technology and expertise, and then they do it as a not-for-profit way of doing it. Because I agree with you, John, we can't charge $5–10 million for one of these treatments, even though you could prove that that's the savings that the state will get, and that is what the amount of money that we've put into it requires for this to at least break even. It's a debate we have to have. And I think hemophilia is going to start that debate because it's the first medium rare disease where patients we know enough patients and enough patients will go on it. And the pricing and reimbursement of that will be a really interesting journey.
Interesting. Okay, so I want to get sure that we can get to at least a couple of the questions that we have from the audience. There are quite a few, but let's start off with this one. How much is the current healthcare delivery system, academic hospitals or others, ready and equipped to deliver gene therapies at scale? What is the missing infrastructure to deliver these therapies? Dave, you want to take that on? Sure.
I mean, I think, obviously this is going to depend on the disease that you're talking about, but already we have a system or a network of institutions that are, when you talk about muscular diseases, that are ready and able to do this, have been participating in clinical trials, now have commercial experience. And I think those, for the most part, get adopted quite quickly. And there's good appreciation of the safety and management of this incident treatment. And I think that these platforms are relatively safe, and if they're well studied and the protocols are well characterized for how you deal with side effects, I think we're capable of scaling these things quite quickly from that perspective.
Anybody else want to take that on?
Well, I would just add, and Dave may know something about Novartis's experiences with Luxturna as well in Europe, but initially in the United States, we chose 10 treatment centers so that we could focus on training the pharmacies as well as the vitreoretinal surgeons involved in the delivery. But I would agree with that, it seemed to me that the training went reasonably well, but you do have to put some thought into it when people have not handled viral vectors before.
Okay. Let's get on to another one. Sandy mentioned some diseases being more accessible to gene therapy than others. Ophthalmology is obviously an area that's seen a lot of gene therapy activity in this regard due to the compartmentalized nature of the eye. What are the specific areas anatomically that the panel sees as having the most opportunity for the next wave of gene therapies? Not the one in the clinic late stage now, but the next stage. Sandy?
I'd love for the brain to be solved for something intravenous. We're getting there. We're almost there. I'd love to be able to do things for the heart, and I'll take lung as my third tissue.
Okay, Jim?
Yeah, I would agree with. I think the heart is a new frontier for the field. CNS is not there yet, at least in any global way.
Okay. Dave?
I agree with the heart. I think, CNS will continue to make progress on. I think the only place I would add is the hematological space. And I think a lot of opportunity still, mainly in ex vivo approaches, but really an opportunity there.
Good. Okay. Kathy?
I don't have anything to add. I agree with the above.
Sounds good. With multiple editing approaches for the same therapeutic indication, for example, ARCUS and base editing for PCSK9 inhibition, to CRISPR versus zinc fingers versus lenti for beta thal sickle cell, beta thalassemia, sickle cell disease, how would patients determine which editing technology is most appropriate for them? It's quite a question. Which one is the safest? How would FDA think about it? I know you've got an opinion on this, Sandy, so I'm not going to start with you. Kathy, how about, well, I'll get to you in a second. But Kathy, how about you? I mean, we are going to be in a situation where patients are going to have to make choices. there are going to be competing therapies out there, and it's a very complex kind of a thing to get started on. And so,
Okay, but that's not new.
okay.
And you know, my own experience in gene therapy is, and I think the same is true for gene editing, when you start to see more than one trial for the same disease, that's a good sign. It means it's working. And you have multiple parties interested in moving it forward. And you know, the answer to what's going to be better, a gene addition approach or a gene editing approach for sickle cell disease, well, you're just not going to know the answer to that until the data are more mature than they are now.
Okay, now Sandy, go.
Well, of course, the answer is obvious. In 10 years' time, nobody will care. This will become such a standard way of doing genomic medicines that you won't look under the hood. I haven't opened the bonnet of my car and worked out what kind of engine it's got. So eventually this will become so standard that it won't matter whether you use zinc fingers or you use zinc fingers or you use zinc fingers.
But John, you know, it is it represents an incredible evolution in rare disease research, where, you know, back in the day, and that's not far, where there were no treatments. It was only diagnosis and go home and enjoy the time you have. And just one anecdotal experience, I attend an annual event with 3,000 parents with Angelman Syndrome, and we go from session, you know, year to year about what could be. And then I was walking out of the last one, and a parent stopped me and I said, which trial should I participate in? There were 2 ASO trials, possibly 2 gene therapy trials. How do I make the decision? And she asked, can I do all of them? I said, no, you can't do all of them. And if you select one of the gene therapy trials, you may not be able to participate in another one. It really is a change, a sea change in the way in which families living with rare diseases have to confront this. And to answer your question about what would be the best editing payload or whatever, if you asked me, I wouldn't know the answer, let alone a patient. I mean, I think it would ultimately be driven by data, but exactly. it is a huge challenge for these small groups, advocacy groups, families, where now they're confronted with all these potential options, where before they were just begging for someone to pay attention to them.
So there's, you all deal with patient populations, I presume, quite a lot, because they're driving this in a lot of different respects, and the unmet medical need is everything. I am curious what your read is of patients' expectations and concerns are right now. Is this something where the patients think that we're at the cusp of seeing a shift towards commercialization of a variety of new gene therapies? Is there concern here that this is going to get bogged down? Are they going to have to wait 5 to 10 years before they can get this? Or is there a sense that this is going to happen sooner rather than later? Dave, how about you?
I mean, listen, I think first and foremost, patients. When we talk about patients, we're often talking about parents, and parents are most concerned about their child and what they can do for their child immediately. I think when we talk about patient advocates, they are concerned about access to these therapies and the rapidity at which that access can be generated. But they've also been through the ringer for decades of failures, and I think many of them do see the promise coming, and they're often have. In many ways, the most noblest patients I've seen in their reflection on how some of the ups and downs of this go. And so while there is often profound disappointment in some of the setbacks that occur, there's also tremendous resilience in those communities. And I know we count on their partnership and rely on them every day for the work we're trying to do. And I can only say that I think it drives everyone who works in this industry to try to fulfill that promise for these folks. And that's the beauty of what we get to work on ultimately.
Well said.
Okay. Anybody want to add to that or subtract? Okay. So the last question I want to get is kind of it's an interesting discussion that has been raised before. The industry till recently used to be known as cell and gene. Do you think that both segments have matured enough for them to be classified as 2 distinct industries? It's kind of an inside baseball kind of a question, but I am curious in terms of your own perspectives on that, how you think that might play out. Is this going to, will we bifurcate the two or continue to put them under the same kind of general umbrella? Anybody?
I think they'll always be together, because I think in many ways the diseases don't care what technology you apply to them.
Yeah.
And I think we can still utilize competing technologies, as we mentioned before, in the same diseases. I think one of the first places this will play out is some of the ERTs, where we have AAV approaches and we have ex vivo approaches being developed concurrently. And we have ERT. And, it'll be interesting to see how that goes. But I think in many ways we will coalesce around these technologies being together, because the delivery, the payload, and these things can be mixed and matched, and the route of administration or the technology utilized will be fungible to the situation that we need to address.
Last round here, just very quickly. I'm curious, looking ahead to 2021, which is the tradition of what we do at JP Morgan, how you see the major things happening in gene therapy, the big events that are likely to happen. How do you see this playing out near term? Sandy?
I think there'll be consolidation of gene therapy with results, clinical results, helping us understand it, and editing will leap forward over it. It's time for editing.
Okay, Kathy?
I hope that we will continue to see products approved in both the US and Europe. So.
Okay, Dave?
I'm hopeful, but a bit pessimistic for 2021. I think we'll see some incremental improvements, but I think we'll continue to see financially staggering disappointments.
Okay, well, Jim?
I would maybe agree with Dave in that there are a number of programs that were started a long time ago that are finally coming to data readouts, and they're likely some of them will fail. That doesn't mean the whole field is flawed. Let's just be aware. I just think we should manage those expectations. And I'm hopeful that this year finally will be in vivo editing readouts, Andy. We look forward to that.
Okay, well, great. Well, we'll keep an eye out for all those things. I really appreciate everybody on the panel joining this discussion. It is a critical discussion. Very interesting time to come together and talk about gene. It's always interesting to talk about gene therapy, but I find that this particular juncture that we're at in terms of the technology and everything out there and the amount of money going into it, the amount of clinical activity, the amount of new research going into new technologies is really fascinating. So thanks for everyone contributing to this conversation. I appreciate the audience taking some time out, and thanks to Ignite for sponsoring this conversation and making it happen. That's it from us for all of JP Morgan. We're wrapped up with this panel, but I really appreciate it again, and hopefully we'll be able to get back together and talk about this later, because it's not over. Thanks very much.
Bye.
Thanks, everyone.
Thanks,
Bye now.
everybody.
Yeah.